Okay, welcome to today's lecture. I hope you're all doing well. My name is Owen Donoghue. I hope you enjoyed the previous lecture on the clinical significance of nucleic acid therapeutics in the context of cancer mRNA vaccines. And today we're going to discuss the next generation of nucleic acid therapeutics. And there's a lot of really exciting technology that's now entering many R&D pipelines. So I'm just going to quickly introduce and talk you through some of these today. And we'll start with self-amplifying mRNA as a potential alternative to mRNA in vaccines. And then, as I mentioned in earlier lectures, we'll also take a look at two emerging genome editing approaches referred to as base editing and prime editing. So we'll start with self-amplifying RNA. So this is somewhat similar to normal mRNA that you were introduced to previously. Just like conventional RNA, it has a 5' cap, a 5' UTR, 3' UTR, and a poly A tail. And most of the sequence in between these features here is protein coding. And it's typically longer than normal mRNA, but the big difference here is that once it's inside cells, this mRNA can make copies of itself before it degrades, whereas normal mRNA can't do this. So after normal mRNA degrades, protein expression from this mRNA stops. In contrast, protein expression from self-amplifying RNA continues and even increases. But this doesn't go on indefinitely, as we'll see later. So it seems like a very useful tool for many reasons, but it's not something that scientists can take total credit for. And this kind of self-amplifying mRNA actually relies on borrowing a few tricks from single-stranded RNA viruses, specifically alpha viruses. So to understand how this works, I think it's useful to quickly look at alpha virus replication. So alpha viruses are single-stranded positive sense RNA viruses. So essentially, alpha virus genomes consist of what looks like a single mRNA. This mRNA needs to contain all the instructions to make new virus and replicate its genome too. And this is all possible with a single mRNA in this case. And it's because the first part of the mRNA encodes what is initially a large single protein. But then when it's translated, it undergoes self-cleavage, generating 4 non-structural proteins referred to as NSP 1, 2, 3, and 4. So one of these is an RNA-dependent RNA polymerase, which just means that it can synthesize RNA using RNA as a template rather than using DNA as a template. But really, all of these non-structural proteins work together to form a few different complexes, and they carry out a variety of functions related to genome replication. So for simplicity, they are sometimes just referred to as the replicase. So first of all, this replicase synthesizes a reverse strand, which acts as a template for further synthesis of a new forward strand. And you end up with lots of new single-stranded positive sense genome being created through this process. And to be brief, all this occurs due to the fact that the replicase recognizes the specific sequences in the 5' and 3' untranslated regions. So it doesn't just replicate any mRNA, it will only replicate RNA that has these features as it preferentially interacts with these. But also, once this double-stranded RNA intermediate exists, the replicase can also utilize what is referred to as a subgenomic promoter to initiate the synthesis of a separate, shorter mRNA from the second half of the genome, using the reverse strand as template. And this mRNA can only be transcribed under these circumstances, and the gene that's on this mRNA can only be translated into protein when it exists on this smaller mRNA, as it's very difficult in the context of the larger initial mRNA for several reasons. Principally as there's a stop codon here upstream of these genes at the end of the non-structural protein genes. So the ribosome will typically disengage from the mRNA at this point here before getting to and translating any of these genes downstream. But if it doesn't disengage here, which can happen sometimes, it could potentially form a fusion protein between these non-structural proteins and the downstream proteins as well, and it's unclear whether that would ever actually be functional. But all of these problems can be resolved nice and easily by just initiating the synthesis of a second smaller mRNA transcript from this point onwards um, after these non-structural proteins. And this can be translated separately without any of these problems. And like the first gene in this viral genome, this second smaller mRNA encodes what is also initially a single protein that also undergoes cleavage to generate separate capsid E3, E2, 6K, and E1 proteins. And most of these go on to form the structure of the new viral particles. And also the new viral genome, which is constantly being regenerated in the background by the replicase, also gets packaged into these new viral particles. And then you have a new fully functional virus that leaves the cell and goes on to infect other cells, and the whole process begins again. However, it was realized that if you delete these genes from the subgenomic mRNA and just replace them with any other gene of interest, for example, in this case, a gene that makes protein X, once the double-stranded replication intermediate is created, you should be able to get the expression of this protein from the viral genome instead of these viral structure proteins. And at this stage, the alpha-virus genome ceases to be a viral genome anymore, because in the absence of any viral structure proteins, there's never going to be any new virus made, as a new protein just can't be used for this generally. But because the replicase protein is still going to be translated from the original longer mRNA, new replicases will constantly be created and constantly recognize the mRNA it came from via the 5' and 3' UTR features, even if it's no longer a viral genome, and even if it's no longer going to be packaged into a new viral particle. And this leads to more and more double-stranded RNA intermediate being generated, and the replicase will also continue to interact with this double-stranded RNA intermediate via the subgenomic promoter, leading to more and more subgenomic mRNA being transcribed, leading to more and more mRNA being translated into protein X. And this is essentially the basis of most self-amplifying RNA technology. And it's a very neat idea. Just to recap, rather than having an mRNA with these standard elements that you learned about previously, if you manufacture an mRNA molecule with these 4 additional alpha virus elements alongside these standard mRNA elements, and you combine them together in this specific configuration, you'll go from having a normal mRNA that is non-replicating to having an mRNA that is self-replicating, giving you this amplification effect. So you can essentially do more with less. So compared to normal mRNA, where the mRNA is delivered to the cell and translated into a protein of interest, where you're absolutely reliant on getting enough mRNA into the cell in order to get high amounts of protein translation, this will stop once the mRNA degrades. This is not going to be the case with self-amplifying RNA. In theory, you just need to get a single molecule into the cell, and this will degrade quickly also. It's not necessarily more stable than normal mRNA, but because in this case, degraded mRNA is constantly being replaced by new RNA, through this self-replication process, you get continued and sustained expression of your protein of interest. But of course, this is not something that can continue forever. Any excessive buildup of double-stranded RNA in the cytoplasm is not going to be something that the cell will tolerate, as this, of course, indicates to cells that there's possibly a virus present, leading to an innate immune response. Even though in this case there's no virus being made, what is happening here still looks very like a viral infection to the cell. And it's important to remember that to a large extent, this foreign double-stranded RNA buildup in the cytoplasm is somewhat limited, at least at the beginning of this process anyways, because in reality, this RNA replication doesn't take place in the cytoplasm. Instead, it takes place in a membrane-bound replication center that's formed by the non-structural proteins. So this double-stranded RNA replication intermediate, in most cases, is actually not accessible, or in some sense, kind of hidden from the cytoplasmic receptors that can detect foreign double-stranded RNA. But this shielding of the double-stranded RNA from these receptors is not always perfect. As you know, in biology, things can be quite messy, and it's hypothesized that if there is enough or high amount of RNA replication occurring, it's inevitable that for many reasons, some of this may escape into the cytoplasm. And once this happens, it can then get detected by these cytoplasmic receptors, leading to an innate immune response. And this response consists of the production of cytokines to attract and alert and stimulate immune cells, which may destroy the cell, or the cell may undergo a programmed cell death itself, also attracting the attention of immune cells and stimulating them to respond to what it thinks could be a viral infection. So this may seem like a bad thing if you're trying to use self-amplifying RNA in cells, but if you recall last week's lecture on the immunology behind cancer mRNA vaccines, and by extension conventional mRNA vaccines, this is actually exactly what you want to happen during any vaccination. First of all, we want antigen expression, followed by the stimulation of an innate immune response. Both of these are essential if you want to have any kind of adaptive immune response initiated, leading to the sampling of antigen by antigen presenting cells, such as dendritic cells, with immune stimulation, leading the dendritic cells then to undergo maturation. Or alternatively, you can deliver this self-amplifying RNA directly to dendritic cells, leading to these cells to also undergo maturation for similar reasons. But both of these paths lead to antigen display on the cell surface of dendritic cells and on MHC molecules, followed by presentation to T cells, as you learned about in last week's lecture. So this is really just to illustrate how self-amplifying RNA can really act as its own adjuvant. In the case of vaccination with normal mRNA that can't replicate, a similar immune response usually occurs in response to the mRNA itself or the LNP delivery vehicle. So in these cases, the cell is also tricked into thinking it's infected, so they can essentially also act as their own adjuvant during vaccination. But with self-amplifying RNA, this self-adjuvant effect in theory should be much more pronounced, as you get much more RNA-based immune stimulation after antigen expression, very much like what happens during a real alpha virus infection. So what's happening in the cells when self-amplifying RNA is present is much more representative of a real viral infection compared to when you just have non-replicating mRNA in cells. Because at this stage, you've many things happening in the cell that would happen during a normal viral infection, except the actual creation of new virus. So when self-amplifying RNA is present, it leads to very specific innate immune signaling that you mightn't necessarily get with other kinds of immune stimulation approaches. For example, like what happens during the use of non-replicating mRNA vaccines. This combination of increased antigen expression and higher quality innate immune stimulation is really useful in the context of kickstarting a long-term protective adaptive immune response against the viral protein, and by extension against the virus itself, or even against cells expressing cancer neoantigens, as you might recall from last week's lecture. So self-amplifying mRNA technology has a really strong potential applications for the vaccine sector. So it's no surprise that the first approved self-amplifying mRNA product is a vaccine. And it's a vaccine against SARS-CoV-2, which was approved recently by the Japanese regulators at the end of 2023. And notably, the dose that was used in this case with the self-amplifying RNA vaccine is very small. It's only 5 micrograms, so it's 20 times lower than the dose that's required with Moderna's SARS-CoV-2 vaccine, and 6 times lower than what's used with the Pfizer-BioNTech vaccine. And it actually outperformed the Pfizer-BioNTech mRNA vaccine in direct comparisons during its clinical trials. And in this trial, it would appear that the antigen expression levels between the Pfizer-BioNTech and the self-amplifying vaccine appeared to be quite similar, but the expression duration was longer for the self-amplifying RNA. So between this and the superior immune stimulation that you get when you use self-amplifying RNA, it makes sense as to why it would outperform the Pfizer-BioNTech vaccine in terms of efficacy. So the obvious and main advantages here with self-amplifying RNA vaccines are mainly centered around this better performance with lower doses, of course leading to decreased manufacturing costs per dose compared to conventional mRNA vaccines. But it's important to remember that this increased immune stimulation that we've talked about here could also lead to increased chances of side effects, such as inflammation. Also, as these amplifying mRNA molecules are much longer than non-amplifying mRNA molecules, it does introduce some extra but not insurmountable manufacturing QC and storage challenges, and this has to be taken into account as well. And some of these things are summarized here in this comparison between self-amplifying RNA and conventional mRNA, which, as always, you can go through yourselves when you have some time. So when it comes to self-amplifying RNA vaccines, there certainly are some pros and cons. One of the most important, I think, that's not discussed enough is the fact that these molecules contain a lot of viral sequence. And from what we understand about alphavirus biology, while improbable, it's not impossible that self-amplifying RNA could recombine with circulating alphavirus genome. And this is because we know this kind of recombination is largely driven by sequence homology. And indeed, we know that in the past, different alphavirus species have recombined in this manner in the field to generate new alphavirus species. For example, Western equine encephalitis virus. So this is something that certainly can happen, and it's something that really needs to be taken into consideration a lot more with self-amplifying RNA vaccines. And this is a problem that we hope to be able to begin working on soon in our lab to see is it something that can be potentially overcome. So I focused on the application of self-amplifying RNA in the context of vaccines here, but it's important to remember that it may also be useful in terms of therapeutic protein replacement or supplementation, like what I talked about a few weeks ago. However, the cellular immune response to self-amplifying RNA, while it's an advantage in vaccination, it's something that's really unwanted in the context of protein replacement or supplementation therapy. And this is because, among other things, this kind of an immune response will reduce protein translation. So there are other groups working towards ways of getting this amplification effect without alerting the immune response of the cell in order to be fully able to explore the full potential of self-amplifying RNA outside its obvious applications in terms of vaccines. So that's self-amplifying RNA, something that's certainly an exciting area. Things are moving very fast with it, and it's certainly one to look out for over the next few years. Moving on, also in terms of emerging technology, a few weeks ago, as you might remember, I introduced the use of CRISPR-Cas9 to make modifications in genomes. And as I mentioned back then, rather than just changing the bases at specific locations, it largely works through the introduction of double-stranded breaks in the DNA, followed by what is sometimes random insertion or deletion of bases rather than making direct changes to the bases themselves. Also, some kinds of more predictable genome editing through this process are possible, but are cell cycle dependent, as I mentioned. But one of the main problems here with the approach that I described a few weeks ago is that it involves making double-stranded breaks in the genome, and these can be quite detrimental if they occur in the wrong place. So a CRISPR-Cas9 based approach that can perform more predictable direct edits in the code in a way that's, I guess, not dependent on cell cycle and not dependent on making double-stranded breaks in the genome would be certainly more desirable. And now there are two ways to achieve this. So the first one I'll introduce is base editing. So the elements of this system are very similar to the normal CRISPR-Cas9 system that I described a few weeks ago. You have the guide RNA and then the Cas9 protein. And this uses a slightly different version of the Cas9 protein, and there are two key differences. Firstly, This Cas9 is not capable of making a double-stranded DNA break. It can only cut one strand, creating what's referred to as a nick. And for this reason, it's referred to as a Cas9 nickase, or Cas9N for short. And the importance of this change will be made clear soon. Secondly, this Cas9 nickase is fused to a deaminase, which becomes positioned in such a way that allows it to preferably deaminate bases located at a fixed position from the PAM site. And you may recall the PAM site from previous lectures. Through this process, deaminases can convert either adenine or cytosine bases to non-standard bases with different base pairing preferences. So between the nicking of the opposite strand and this deamination, you can convert certain base pairs to certain other base pairs, depending on what kind of deaminase you use. To take a closer look at how this works, we look at the action of an adenine deaminase Cas9 nickase fusion protein system, which is referred to as an adenine base editor. So first of all, after binding of the CRISPR guide RNA, the nickase and deaminase are brought into their positions, making a single-stranded nick and an adenine deamination in the expected locations. So the single-stranded nick initiates a natural cellular DNA repair process, and as this nick always occurs on the strand opposite the deamination, this non-cut strand, and by extension the base pairing preference of the deaminated base, is used as template for the repair. So through this process, a precise change can be introduced in the nicked strand, and eventually in the other strand, another change can be introduced through either further DNA replication or DNA repair, and the deaminated base is eventually replaced by a new base, and this new base that is inserted in this case will be a guanine due to the presence of a new cytosine on the opposite strand. And this is how you can use base editing to convert an AT pair to a GC pair. And of course, using a cytosine base editor, you can introduce other edits in a similar way. However, this involves the introduction of a uracil after cytosine deamination. And this is something that the cell does not really tolerate in DNA. So this deamination is quickly excised by cellular uracil DNA glycosylase. So to counteract this, typically cytosine-based editors also have a second protein fused to the Cas9 nickase that acts as a DNA glycosylase inhibitor. So if you're ever wondering why you see this extra component with a cytosine-based editor architecture, this is why. It's also important to remember that the actual base that becomes deaminated is in fact just the base that is most likely to become deaminated based on the configuration of these fusion proteins after the incorporation of the guide RNA. So in other words, other bases in close proximity inside a so-called editing window can also get edited. So the base that is preferably edited and the size of the editing window and the location of the editing window relative to the PAM site will vary from one kind of editor to the next depending on the architecture of the fusion protein. And there's a lot of useful diversity out there in terms of these properties across the diverse range of cytosine and adenine base editor architectures that have been developed. A lot of this is summed up really well here in this figure taken from this publication. It's a bit hard to see the detail here, but you can always check it out yourselves by going to the publication. But in all cases, in this figure, the editing windows for the cytosine and adenine base editors are highlighted in green and red respectively, and the PAM site is at the extreme right of each of the sequences. So you can see how much flexibility there is in terms of the positioning of these editing windows with different base editor designs. But although there may be some diversity in terms of editing windows, it might not be enough in some situations, as this approach may not be suitable for all targets. And also within editing windows, it's still possible to get a lot of off-target editing taking place. Also, it's difficult to make several changes at the same time. But another type of editing technology has emerged that has introduced an awful lot more flexibility into this process. And this process is referred to as prime editing. And again, this involves the use of a Cas9 nickase, but this time it's fused to a reverse transcriptase, As some of you may know, this is an enzyme that can be used to synthesize DNA strands using RNA as a template. And it's a little trick that's been borrowed from retroviruses. So, like other systems that I introduced, it also requires a guide RNA to guide the Cas9 fusion protein to the target region. But this time, the guide RNA looks a little bit different, of course, and you'll see why soon. And it's referred to as a prime editing guide RNA, or PEG RNA for short. So let's take a look at how this works. So in order to explain this, rather than making a diagram myself, I looked around for what I thought might be a good diagram to use to explain this process. And the best one I could find was this one here from Wikipedia. So I've adapted it slightly for this. So we'll begin here in step 1, where we see that, like the other systems that I've introduced, the guide RNA binds to the target DNA sequence in the genome. But the Cas9 nickase used here in this case nicks the opposite strand, not the binding strand. So it makes a nick on the same strand as the PAM sequence, very close to the PAM sequence. And this cutting modification is important because the 3' end of the PEG RNA is complementary to the genomic DNA regions that are on the 5' side of the cut. And once this cut is made, here it can compete with the genomic DNA strand for binding to this region. And if the PEG RNA binds to this DNA, the 3' end of the DNA then acts as a primer for the reverse transcriptase. And where it binds to this is sometimes referred to as the primer binding site. So once this happens, the reverse transcriptase begins to extend the DNA strand from this paired 3' end using the adjacent single-stranded PEG RNA sequence as a template. And this RNA template is similar to the genomic DNA sequence on the 3' side of the nick, but with the exception of some base changes that are here in yellow. And it's these base changes in yellow that represent the changes you want to make to the genome, and they end up incorporated into the newly synthesized DNA strand. So this newly synthesized DNA strand and the original DNA strand form two flaps, one edited and one not edited. And these two flaps compete for base pairing in this region of the genome. And of course, this flap situation is not something that's going to be tolerated by the cell. So only one of these flaps ultimately gets reincorporated back into the DNA in the long term, and the other flap is excised. And this occurs through the cell's own DNA repair mechanisms. So if the 3' flap is reincorporated back in and the 5' flap is excised, of course, this is more ideal in this case, as it means that the mutation has a chance of remaining permanently. And as the mutations, again here in yellow, are detected as mismatched bases by the cell, it will try to repair this mismatch. And in this case, it will do either one of two things. It will either use the mutated strand as template, or it will use the non-mutated strand as template for the repair. And if it uses the mutated strand as template, then the mutation stays. And it may be possible to shift the template strand selection in favor of the mutated strand by using another guide RNA to make a nick in the non-mutated strand. But this could potentially lead us back to a situation where we may have blunt or staggered double-stranded breaks, which may be better to avoid for safety reasons. So that's prime editing, and it's an approach that represents, I think, quite an innovative advance, giving us a way to make very specific and predictable changes in the genome by simply programming them into the guide RNA and getting them incorporated then into the genome. And with this, there's a lot more flexibility to introduce more complex changes compared to base editing without any unintended bystander mutations. So it really gives you more control over the potential outcome, which is always a good thing in these situations. So there are clearly lots of advantages to be had here with prime editing. But both approaches have their advantages and disadvantages, and these are outlined here. As usual, you can go through this table yourselves. And while prime editing has many advantages over base editing, at the moment the editing efficiency with prime editing is still a big issue. This makes sense when you think about it, as you can see towards the latter stages of the prime editing process. You really are reliant on certain events going your way in order to get the desired changes incorporated into the genome. And in terms of clinical programs with either of these approaches, as they're both quite new, there's not a lot of phase 1 trials that have been initiated to date. But a good example of one of the main companies that is active in base editing clinical programs is Beam Therapeutics, uh, which have several phase 1 trials underway for several inherited conditions and have a separate preclinical program engaged in the use of base editing in the development of off the shelf CAR T cells. There hasn't been as much movement in the prime editing space, potentially because the technology itself is quite expensive to license from Prime Medicine. But recently, Prime Medicine got FDA approval to commence with their own phase 1 trials involving the use of prime editing to treat CGD. So we'll see what happens with these. There's bound to be more activity in this space as time goes on because I really think these two technologies in particular represent two big transformative leaps in terms of genome editing technology, paving the way for much more precise, safer, and versatile methods of genetic modification. And it's reasonable to expect that there'll be a huge expansion in these particular R&D pipelines as time goes on, because all of a sudden we're at a point where there's a lot of things that have really now become feasible to do because of the existence of this type of technology. So it's certainly an interesting and exciting time to be involved in the field. And this brings us to the end of today's talk. I hope it gives you some kind of an insight into some of the important emerging technology related to nucleic acid therapeutics. So take care and see you soon.